So this lecture we are going to study about affinity chromatography. This is a very famous type of chromatography, and most probably this is the last stage of protein purification, because this will give a clear refinement in your protein purification, and you will get exactly your protein of interest, and without any further the load of any other proteins. So that is why this type is very important. And unfortunately, the preparation of affinity chromatography media, that means affinity resins, it is a little expensive. So you have to maybe find a methodology, and you have to attach the type of the molecule going to be interacted with your protein of interest, that has to be attached with the resin, either by directly to the resin, or otherwise with a carbon arm, to facilitate the binding process may be a little flexible. So this type of method is very important, at the same time little complex when compared to any other type of chromatography. And this affinity chromatography is a separation technique. You can see here, this is a technique, and this is separation technique with many applications, since it's possible to use it for the separation and purification of most of the biological molecules, the basis of the function, that is based on the chemical structure. For example, if your biological material is enzyme, you can use a substrate, or you can use inhibitor. And if you want to separate the antibody, you can use the antigen. If you want to separate the antigen, you can use the antibody. So based on that, we can select. So these interactions are very specific. For example, this enzyme and substrate means one enzyme will interact to only one substrate. One enzyme will interact with only one inhibitor. So this antigen antibody is also 100% very specific. One antigen will bind only with the antibody. So this is not, there is no cross binding, or otherwise any other non-specific binding will not be possible. So that is why this type of purification is very famous and very specific binding, and that is why you can specifically purify your protein of interest. The molecule to be purified is specifically absorbed. For example, a cell lysate applied to the affinity column by a binding substance, that means the ligand, so which is immobilized on an insoluble support, that is a matrix. Matrix is insoluble, and that is immobilized means that can be attached. The ligand can be attached to the matrix. I'll explain to you with the practically more detailed one. You can see here, this is the resin. So this resin we are going to attach with an antibody. Here these are all the antibodies are going to be so we are going to purify the protein, that is the antigen. So this antigen can be separated. How? Because this is the resin, the matrix, and we attach with antibody, that is ready made. Just you can pack the column, then you can load the sample with the pH 7 buffer, and the protein recognized by the antibody. That means this protein is recognized by the antibody, means this your type of antigen is going to be recognized. This antigen antibody interaction is very specific. Only this one will bind, and you can see here the blue color one, the protein recognized by the antibody. That means this one cannot be bound. So this one is very specific, so that is why. And you can separate specific molecules, and the specific molecules can be separated by wash, then the removal. And so this is the principle, just you can have a look.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application.
I'm explaining the stages. Now the sample application wash, that means so this is the ligand, this is spacer arm, and this is the matrix, and this one is very specific to your protein. So if you are applying the sample, only this area, okay, this is called as a ligand. So this ligand will attach to your protein only. For example, this is not your protein. So this interaction, this red color one and this arm, okay, this is a ligand attached with the matrix by a spacer. Okay, this spacer will facilitate the more space to be interacted. For example, if you are attaching, okay, this the cone shape directly to the matrix, so there is no space for interaction. That is why we have to give the space for interaction. That means, so this is a carbon arm. Okay, this carbon arm facilitates the more flexible binding of this ligand to your protein. So this is not your protein. Just you can have a look. I'm going to play
And wash. The goal in this step is to bind the target molecules and wash out all unbound.
You can see this is your protein, the green color one. So this is having some interaction area. That means if it is an enzyme, this may be the inhibitor. If it is an antigen, this may be the antibody. If it is an antibody, this may be the antigen. If it is an enzyme, okay, this may be the substrate. But you have to ensure that if you are using the substrate molecule can be hydrolyzed. Okay, so that is very, very important. Okay,
material.
You can see here this some of the
non-binding molecules will pass through in the flow-through during washing with binding buffer.
So here your protein of interest, okay, this one can be specifically bound to the ligand molecule, and no other molecule will be there in the column. Just you can have a look here. So all the unbound molecules is going to come out.
In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by.
So this is elution method. The type of elution, just you can use the same concentration of the ligand molecules, and the ligand molecule will bind directly with this molecule as a competitive binding, and automatically this may be detached, okay, from the matrix, and you can get it. Okay, this maybe you can use either the same ligand, the free ligand concentration to liberate, okay, this one. This again, the free ligand can be separated from your molecule by dialysis. So this is the way. Okay,
change in the buffer composition. A common way is to increase pH of the buffer.
So there are two ways of elution. One is by changing the pH of the buffer, or otherwise you can use the concentration of the various concentration of so your ligand molecule, free ligand molecule. You can use the free ligand molecule, this one. Simple method is the changing the pH. pH means the same buffer, you can increase the pH range. Okay, that means the pH can be slightly increased. Okay, that may be interacting with the molecule. That means the protein molecule interacted with the ligand. Okay, this interaction may be disturbed. Okay, that is why the protein can be liberated. But sometimes you have to be very specific that the pH should not denature your protein. If you are using a particular pH, and that pH can detach your protein from the ligand, at the same time that protein should not be denatured or degraded, or its activity should not be lost. Okay, your main intention is the purification, and the purification should not completely denature your protein. So that is why this is very, very important, and this type of affinity chromatography is very famous. And my advice to you, okay, that is you can use always the concentration of the free ligands, so that is safe, and at the same time you cannot lose the enzyme activity. And if you want to purify any enzyme, if it is a protein molecule, there is no problem. Okay, maybe you can change the pH. Like this method, they already explained about that. This is the pH change, that is this is the neutral pH to the higher pH level, and this molecule can be removed, and automatically the pH can be normalized. The problem is you can get the molecule by elution. At the same time, this molecule should not be degraded or otherwise denatured. So this is a major step, and you have to be very careful in that. Okay, you can study the molecule whether the pH is going to denature your protein or not. If it is not denaturing it, then you can go with the pH change. Or if it is denaturing your protein, means just you can go with the alternative method, that is addition of, okay, this free ligand molecule. Okay, so this is a video. So this is from Sativa. You can see a lot of videos, okay, in this website. Okay, just you can have a look, and this type of informations are very useful to you. And this is a very famous company, Cytiva, and they have a lot of videos. It's open, anybody can watch. Yeah, this is the pattern of affinity purification. Just you can see here, this is the absorption by 280. That is mostly the proteins will absorb, okay, this maybe the wavelength, and this is the ml, how many ml is used for the fraction, and this is the binding buffer. Okay, just you can see here, this is after loading, the unbound molecules is going to be liberated, okay, from the column. And elution buffer, that means change the pH. After change the pH, automatically you will get eluted target protein, only one peak. So no other cross contamination or otherwise no other peak will be there, only one peak, that is your protein only going to be eluted. Because you should know in this aspect, that means your protein, what is the ligand you are going to choose it. If you are choosing the ligand, so that is very specifically interacting to your own protein, the protein of interest, that is okay. And if it is a common one, maybe for some other enzyme, for example, I will say an example of the glucose. The glucose is a substrate for two enzymes, one is glucokinase and hexokinase. If you want to purify a glucokinase enzyme, if you are using, for example, if you are ligating, or the ligand is a glucose molecule, what will happen? The glucose molecule not only attracts a glucokinase, at the same time the hexokinase can also be. So that is why. So you will get here the two peaks, or otherwise within the peak, okay, two molecules will be there. So the selection of ligand is very, very, very important. And if you are choosing wisely, exactly, okay, this specific interaction of your protein of interest, and you will get exactly 99% purity here, and based on your technical skill, and you will get the highly purified fraction. So you will get only the single band in the SDS page, okay, and with the silver staining also. So this is a very famous one. I'll play the video of affinity animation of the old video of Amersham Biosciences. Just to have a look. So this is the principle of affinity chromatography. So the separation depends on the reversible adsorption. Reversible adsorption means the binding of biomolecules through biospecific interaction. It is not the chemical interaction, it is biospecific interactions on the ligand. This is having three main stages: equilibration, sample application wash, and finally elution. See here. So this is the ligand, and this is space RAM. A biospecific ligand can be attached, and the first one is equilibration. So the first step is equilibration of the stationary phase of the desired start condition. That means you can equilibrate with your own buffer that will stabilize the molecule of the ligand with the space RAM. Next one is sample application. Now you are going to apply the sample, the mixture of samples. Unbound molecules are coming out, okay, from the resin. You can see here only the specific interaction is retained. That means the molecule is going to be interacting with your ligand here. This is your ligand, and this is the protein molecule of your interest. So this is only retained, and no other protein is present. I am repeating again, I am loading the mixture of proteins. So this will not bind because there is no interaction site, and this will bind because this is having the specific interaction. And usually this one will also not bind to the resin because there is no specific interaction. There is no binding site here, and here also going to be eluted. Okay, this one is this washing step removes all the unbound molecules. So this is the washing step. So that is why absorption will be increased, okay, by washing. After washing, you can see here only your protein will be bound with the resin. And the next one is elution. So change the pH, and automatically due to the change of the pH, automatically all the ligands liberate, or otherwise the interaction between the ligand is I'm repeating again. So we are going to apply, the blue color is the pH change. Due to the pH change, this interaction is going to be detached, so you will get the fraction of your own protein, and whatever you are going to get it, okay, that has to be dialyzed with the normal buffer, because the pH change should not disturb your protein or denature your protein immediately after getting the fraction, and you have to dialyze it, okay, to normalize the pH of your protein. And this is the animation. Maybe I hope you can understand the concept of affinity chromatography. This is a very important question for the examination also. And this purification may be it's a very high level of purification, and the protein purification fold can be excellent, okay, in this type of purification. So this method can however be quite costly, okay, when compared to other types, and this is time consuming also, because you have to prepare the affinity methods, that is affinity ligand preparation and the spacer arm, and if you are standardizing it, and you can use it for the multiple times, no issues at all. The affinity chromatography, okay, was used initially for the protein isolation and the purification, and particularly enzymes, okay, can be used. And after that, that can be used for the purification of monoclonal antibodies, and that is very specific to the antigens, so that also can be separated by using this one. The enzyme inhibitors can be used, the antibodies used for the separation of antigens, interferons can be used, and recombinant proteins with small scale purification of nucleic acids. So all the different types, so now it is diversified, okay, that means affinity chromatography now used for mostly all the applications, but the specific interaction is recommended. And next type of reverse phase chromatography that we can see in the next class.